Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to be covering chapter 11 for our MCAT Physics and Math playlist, and this chapter is titled Reasoning and Research. In this chapter, we're going to cover the following objectives. First, we're going to go over the scientific method and the finer method. Next, we're going to discuss basic science research and go over concepts like controls, causality, and identifying potential sources of error that can influence experimental outcomes. Following that, we're going to dive into human subjects research, where we're going to compare the experimental approach with the observational approach and also address common error sources in these studies. After that, we're going to look at the principles of ethics in research, and then we're going to discuss the importance of these principles in conducting ethical studies. Finally, we're going to explore research in the real world. So here, we're going to learn how to differentiate between populations and samples. We're going to define generalizability, and we're also going to understand how research findings can support intervention. With this introduction, let's go ahead and jump into Objective 1, where we're going to explore the scientific method and the finer method. To help bring these concepts to life, we're going to use a simple experiment involving light and plant growth as our example. This example will guide us as we break down the steps of the scientific method and evaluate how the finer method ensures a research question is both practical and impactful. By following the scientific method, researchers ensure that their work is logical and reproducible, and that helps build confidence in the conclusions they draw. The scientific method is broken down into 8 essential steps. First, scientists generate a testable question. So this step begins with curiosity about a specific observation or phenomenon. For example, if a researcher notices that plants grow differently under certain light conditions, then they might ask, does the color of light affect plant growth? This is a good testable question because it is clear, it's specific, and it's measurable, which means it can be addressed through experimentation. Next, they gather data resources. So this involves reviewing existing research or literature on the topic. For instance, before conducting their experiment, the scientists might look at previous studies on photosynthesis to understand what's already known and then identify gaps in that knowledge. This step makes sure that the study builds on prior work instead of just copying it. The third step is to form a hypothesis. This is essentially an educated guess that's based on the information gathered in step 2. A hypothesis is often structured as an if-then statement. So if plants are exposed to red light, then they will grow taller than the plants exposed to blue light. So this statement provides a clear prediction that can then be tested through experimentation. After forming a hypothesis, the next step is to collect new data. This is where the researcher designs an experiment to test their hypothesis. So they might grow identical plants under different light colors and then measure their heights over time. During this step, careful control of variables is super important so that you can isolate the effect of the light color. So the scientist needs to make sure that all of the plants get the same amount of water and the same amount of nutrients so that they can truly observe the relationship between light color and plant height. Once the data is collected, now it's time to analyze it. Researchers can use statistical methods to determine whether the results support or refute the hypothesis. And they might calculate averages and look for significant differences in plant heights across the light treatments. This analysis helps turn that raw data into meaningful conclusions. The sixth step is to interpret the data. So here, scientists compare their findings to the original hypothesis and then assess whether it was correct. If the hypothesis is supported, that's great. But if not, it doesn't mean that the research was a failure. It just means that the hypothesis needs to be revised or the experiment needs to be redesigned. And that's okay because science is an iterative process, and sometimes unexpected results often lead to new insights. The next step is to publish the findings. Sharing results with the scientific community ensures transparency, and then it allows other researchers to build on that work. For example, the plant study might be published in a journal where other scientists can review the methods and then even repeat the experiment to verify the findings. And that actually leads us to our final step: verify the results. Reproducibility is a cornerstone of science. So if other scientists can repeat the experiment and achieve similar results, then that strengthens the confidence in that study's conclusion. Now, alongside the scientific method, researchers often use the finer method to evaluate the quality of their research question. The finer method makes certain that the question being asked is meaningful and practical to investigate. Let's break down the criteria. The first criteria is feasibility. Researchers have to ask themselves whether the study can realistically be conducted with the available resources, time, and technology. For example, while it might be fascinating to study plant growth on Mars, it's not feasible without access to space travel. The second criteria is interest. A good research question should capture the interest of the scientific community. If the question is engaging, it's more likely to inspire collaboration and further investigation. Next, the question must be novel. It should address something new or provide a unique perspective. So studying the effects of red light on plant growth might be novel if no one has specifically examined how it impacts root development. The fourth criteria is ethics. All research must adhere to ethical standards, especially when it involves human or animal subjects. Researchers have to consider factors like informed consent, minimizing harm, and respecting the rights of participants. Ethical guidelines make sure that the pursuit of knowledge doesn't come at an unacceptable cost. Finally, the question should also be relevant. This means that the research should have practical implications or contribute to broader scientific understanding. So, for example, a study on light and plant growth could be highly relevant for improving agricultural practices or indoor farming. So, by combining the systematic steps of the scientific method with the evaluation framework of the finer method, researchers know that their work is not only well structured, but it's also impactful. With that, we can move into Objective 2 and discuss basic science research, which is often considered the easiest type of research to design. This is because the experimenter has the most control over the variables and conditions. Basic science research focuses on understanding fundamental processes by carefully manipulating variables to observe specific outcomes. At the core of any research experiment are independent and dependent variables. The independent variable is the factor that the experimenter changes or manipulates, while the dependent variable is the outcome being measured. For example, in our ongoing light and plant growth experiment, the independent variable could be the color of light, red, blue, or green, and the dependent variable would be the plant height after a set period of time. To ensure that the results are meaningful, researchers use controls, which are elements of the experiment that remain constant, to isolate the effect of the independent variable. Controls can be divided into two main types: positive controls and negative controls. Positive controls are designed to confirm that the experiment can produce a measurable effect. For instance, if we already know plants grow well under white light, including a group exposed to white light would serve as a positive control to validate our methods. Negative controls, on the other hand, ensure that there is no effect when the independent variable is absent. So in our experiment, a negative control could involve growing plants in complete darkness to demonstrate what happens without any light exposure. Another key concept in basic science research is causality. This establishes an if-then relationship between the independent and dependent variables. So, for instance, if the hypothesis is if plants are exposed to red light, then they will grow taller, causality would require that the change in plant height is directly related to the color of the light without interference from other factors like water, soil, or temperature. However, even with well-designed experiments, error sources can affect the validity of results. These errors generally fall into two categories: systematic error and random error. Systematic error refers to consistent inaccuracies that skew results in a particular direction. These errors often stem from flaws in the experimental setup, such as a miscalibrated ruler that might consistently measure plant heights too short. Random error, on the other hand, results from unpredictable fluctuations, such as slight differences in environmental conditions that vary from day to day. Random errors are usually less concerning if the sample size is large enough because their effects average out over multiple trials. To further understand error, we can break it down into two related but distinct concepts: precision and accuracy. Accuracy, also called validity, refers to how close a measurement is to the true value. So for example, if the actual plant height is 10 centimeters and our measurements consistently hover around 9.8 to 10.2 centimeters, then the experiment is accurate. Precision, also called reliability, this refers to how consistent repeated measurements are, even if they aren't close to the true value. If every measurement of plant height is exactly 8.5 centimeters, then the experiment is precise, but it's not accurate. Now, this target analogy really helps in remembering accuracy and precision. So this target here in the upper left corner, this describes a scenario where the results are accurate, but not precise. So you notice that the data points are scattered around the bullseye, right? But they are a little bit inconsistent between measurements. So the data points are scattered around the bullseye. They're close to the true value on average, but there is little consistency between measurements. The target In the upper right corner, this represents a scenario where the results are both precise and accurate. So here, the data points are tightly clustered around the bullseye, and this shows both consistency and closeness to the true value. This is the ideal outcome in research. Now, the target in the lower left corner, this illustrates a situation where the results are neither accurate nor precise. So the data points are very scattered and they're far from the bullseye. So this indicates that it's inconsistent and inaccurate. Finally, the target in the lower right corner, this depicts a scenario where the results are precise but not accurate. The data points are tightly clustered, but they're far from the bullseye, and that shows consistent measurements that are systematically incorrect. So this is often due to systematic error. And with that, we've completed the second objective, and now we can move to objective 3, where we're going to discuss human subjects research. This type of research presents unique challenges compared to basic science research. Because it involves real people, human subjects research has to account for ethical considerations and the complexity of human variability. Now, we're going to start by discussing the experimental approach, which is commonly used in biomedical research. In this approach, researchers manipulate an independent variable to determine its effect on an outcome while carefully controlling for any external factors. 3 key elements of this approach that help ensure reliable results is randomization, blinding, and data analysis. Randomization is a method that's used to evenly distribute confounding variables between groups. For example, in a clinical trial testing a new medication, participants are going to be randomly assigned to either the treatment or the control group. This reduces the risk of unintentional bias, such as differences in age, gender, or pre-existing conditions that might affect the results. Randomization ensures that any observed effects are more likely to result from the treatment itself rather than any unrelated factor. Next, blinding is used to remove bias by limiting what subjects and researchers know about the treatment groups. We can have single-blind or double-blind studies. In a single-blind study, participants do not know whether they're receiving the treatment or a placebo. This helps minimize the placebo effect, where participants experience perceived benefits simply because they believe they're receiving the treatment. In a double-blind study, neither the participants nor the researchers know who is in what group. So this additional layer helps reduce the potential for researcher bias, where expectations could unintentionally influence data collection or data interpretation. The final component for the experimental approach is data analysis. This is where researchers use statistical techniques to evaluate their findings. For example, researchers might use regression models to adjust for potential confounders like age or baseline health differences between groups. So these kinds of methods really help ensure that the results are valid and that they provide meaningful insights into the treatment's effectiveness. Now let's shift to the observational approach. This is used when experiments are not feasible or ethical. Observational studies do not involve direct manipulation of variables, but instead they examine relationships between exposures and outcomes. There are 3 main types of observational studies in medicine. We have cohort studies, cross-sectional studies, and case control studies. In a cohort study, researchers follow a group of individuals over time to assess the rate of an outcome based on exposure to a factor. So they might track a group of smokers and non-smokers over 20 years to compare lung cancer rates. These studies are valuable for establishing temporality because they observe events as they unfold. A cross-sectional study, on the other hand, assesses both exposure and outcome at a single point in time. For example, a study to determine the prevalence of lung cancer in smokers and non-smokers at a given point in time would be an example of a cross-sectional study. Finally, case control studies look retrospectively at exposure history by comparing individuals with a specific outcome to those without it. For example, a study might examine past smoking habits of individuals with lung cancer compared to those without. Case control studies are really useful for studying rare outcomes. When evaluating these observational studies, researchers use Hill's criteria to determine whether an observed association is likely to be causal. So here we have the criteria, and we want to go over all 9 of them. Just to restate, Hill's criteria describe the components of an observed relationship that increase the likelihood of causality in the relationship. These criteria do not provide an absolute guideline on whether a relationship is causal or not. So for any observational study, the relationship should be described as a correlation. Now let's start with that first criteria, temporality. This states that the exposure must occur before the outcome. For example, smoking must precede lung cancer for it to be considered a cause. The second criteria is strength. Stronger associations provide greater support for causality. The third criteria is dose-response relationship. As exposure increases, the likelihood or severity of the outcome should also increase. So for instance, heavier smoking is associated with a higher risk of lung cancer. The fourth criteria is consistency. So the association should be observed across multiple studies and in different populations, and that provides confidence in the findings. The fifth criteria is plausibility. There should be a biologically reasonable mechanism that links the exposure to the outcome. The sixth criteria is specificity. A specific exposure should lead to a specific outcome. So for instance, we know that asbestos exposure is closely linked to cancer. The seventh criteria is coherence. The association should align with pre-existing knowledge. For instance, there's evidence of smoking's harmful effects, and that's consistent with what we know about carcinogen exposure. The eighth criteria is experiment. When possible, experimental evidence strengthens the causal link. And then finally, we have consideration of alternative explanations. Researchers must rule out other factors that could explain the observed relationship. For example, genetic predispositions is a good one. Now, considering all this, we can move into our discussion on the error sources that can arise in human subjects research. These errors can distort results, and then they can lead to inaccurate conclusions if they're not properly addressed. Now, we're going to start with selection bias. Selection bias occurs when the sample is not representative of the population being studied. For example, recruiting only healthy volunteers for a study on chronic illness can skew the findings and then limit their generalizability. Next, we have detection bias. This arises when researchers inconsistently search for outcomes across different groups. For example, if healthcare professionals are more likely to screen smokers for lung cancer than non-smokers, then the study might overestimate the prevalence of lung cancer in smokers. Third, we have observation bias, also known as the Hawthorne effect. This occurs when participants change their behavior because they know they're being observed. For instance, participants in a diet study might eat healthier while they're being monitored, and that can distort the results. Finally, confounding happens when a third variable is associated with both the exposure and the outcome, and that can lead to a false or misleading association between the two. For example, imagine a study investigated whether ice cream consumption is linked to a higher number of drowning incidents. At a first glance, the data might show a positive association between these two variables. However, this relationship is confounded by a third variable, and that third variable is hot weather. Hot weather increases both ice cream consumption and swimming activity, which in turn raises the risk of drowning. So in this case, the association between ice cream consumption and drowning is not causal. It's the result of the confounding effect of temperature. With that, we have completed the third objective. Now we can move on to objective 4, where we're going to discuss the core ethical principles that guide both medical practice and research. Ethics plays a fundamental role in making sure that all actions in medicine and research respect the rights and well-being of individuals while promoting fairness and minimizing harm. In medicine, there are 4 core ethical tenets. The first one is beneficence. This is the obligation to act in the best interest of the patient. For example, a physician recommending a life-saving surgery, even though it involves some risks, fulfills the principle of beneficence because the overall goal is to improve that patient's health and well-being. The second tenet is non-maleficence. This is often summed up as do no harm, and this principle requires avoiding treatments or interventions where the potential for harm outweighs the potential benefits. For example, a doctor might avoid prescribing a medication with severe side effects if there is an equally effective alternative with fewer risks. The third tenet is autonomy. This emphasizes the responsibility to respect a patient's decision about their own healthcare. For example, if a patient declines a specific treatment, even one that could be life-saving, their choice must be respected. So this is a really important tenet. Autonomy underscores the importance of things like informed consent, where the patient fully understands the risks and benefits before making decisions, and then when they make their decision, healthcare providers have to respect that. The fourth tenet is justice. This principle involves treating similar patients with similar care and then distributing healthcare resources fairly. For example, during a shortage of donor organs, justice requires that organs be allocated based on medical need and the likelihood of benefit rather than non-medical factors like socioeconomic status. While these 4 tenets guide clinical medicine, research ethics relies on a slightly modified framework established by the Belmont Report. This report outlines 3 key ethical principles for research involving human subjects. They are respect for persons, justice, and beneficence. So let's look at these in detail. Respect for persons. This principle includes respecting autonomy, ensuring informed consent, and protecting the confidentiality of participants. So, for instance, in a clinical trial, participants must be provided with all of the necessary information about the study, including its purpose and the risks and benefits, so that they can make an informed decision about whether to participate. In addition, researchers must take extra care to protect vulnerable populations like children or individuals with cognitive impairments. The second principle is justice. In research, justice dictates which study questions are worth pursuing and how study subjects are selected. So, for example, researchers must ensure that the benefits of a study are distributed fairly across all populations and not only to those who participated in the study. So, justice also prevents the exploitation of vulnerable groups. Finally, beneficence is the principle that requires researchers to maximize potential benefits while minimizing harm. So by adhering to these ethical principles, both researchers and medical practitioners can ensure that their work respects human dignity, promotes fairness, and contributes meaningfully to society without causing any undue harm. Now, we move on to our last and final objective. Here, we're going to discuss how research is applied in the real world. Let's start by first discussing populations and samples. A population includes all individuals who share a set of characteristics. So, an example of a population could be all of the adults with diabetes in a particular country. When we study populations, the measurements we calculate are called parameters. However, studying an entire population is often impractical due to the size and/or accessibility. Instead, researchers use a sample. This is a smaller representative subset of the population. Measurements from the sample are referred to as statistics, and they're often used to estimate the parameters of the larger population. Now, the next concept we want to discuss is generalizability. And in order to understand that, we need to talk about internal validity and external validity. So, internal validity refers to the extent to which a study accurately identifies a causal relationship between an independent and dependent variable. For example, in a study testing a new medication for diabetes, strong internal validity means that changes in blood sugar levels are directly caused by the medication and not by other factors like diet or exercise. Achieving internal validity requires rigorous control of variables and careful study design. External validity, often referred to as generalizability, refers to how well the results of a study can be applied to the broader population. For instance, if the study only included participants from one geographic area, then the findings might not generalize well to populations in other regions. Achieving external validity actually requires a representative sample and considering diverse population characteristics. Now let's discuss statistical significance versus clinical significance. Statistical significance refers to the likelihood that the observed results are not due to random chance. For example, if a diabetes medication lowers blood sugar levels by a small amount, but the p-value of the results is less than 0.05, then the findings are statistically significant. However, this doesn't necessarily mean that the results are going to be impactful in practice. Clinical significance, on the other hand, refers to whether the intervention has a meaningful effect on a patient's outcome. So, for instance, while that new diabetes medication might statistically lower blood sugar levels, if the reduction is so small that it doesn't actually improve the patient's quality of life or reduce complications, it would lack clinical significance. So, a truly impactful intervention must provide results that matter both statistically and practically. And with that, we've completed this lecture. I hope that it was helpful. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.